This presentation is entitled Managing Invasive Pests in an Agriculturally Diverse State. Florida has many ports of entry used for international commerce and receives millions of international visitors each year. Many of these visitors are arthropod pests that are unintentionally brought into the state. Florida is comprised primarily of tropical and subtropical growing environments that provide habitat for pest organisms year-round. Florida is an extremely diverse state in terms of the crops that are grown. Watercress is a lesser-known crop grown in Florida. It is an aquatic crop in the Brassicaceae, the same family as cabbage and broccoli. In 2014, a mysterious new disorder was observed affecting watercress, causing dieback of the crop and tissue necrosis. A previously unrecorded leafhopper was found infesting affected watercress that year. It was identified as Macrostyles near-severini, which transmits a phytoplasma causing the necrosis and dieback observed. The causal agent impacting watercress was confirmed by the UF Diagnostics Lab to be watercress phytoplasma, belonging to the aster yellows group. The first step when a new invasive pest is identified is to determine its host range in order to understand which crops may be impacted. The watercress leafhopper is originally from California, and it established in Hawaii in 2002. This slide includes a table of some common weed hosts of the watercress phytoplasma that were identified near watercress farms in Hawaii. Each of the weeds listed in the table is also present in Florida. This is not a comprehensive list, but is an example of the sort of information that must be gathered when a new pest arrives in the state. In addition to ecological information, such as host range, growers need information on management, and in some cases, new management tools. In collaboration with the IR4 program, field trials were designed on a large watercress farm impacted by the watercress leafhopper soon after its establishment. The two insecticides listed in green, Advise, or imidacloprid, and Movento, or spirotetramat, were registered for use on watercress and were being used by the grower to manage the new pest. However, there are limits to the number of times each insecticide can be applied to the crop, and overuse of any type of insecticide can lead to the development of resistance. 5 other insecticides, none of which were registered for use on watercress, were evaluated in the spring of 2014 in a replicated on-farm field trial. 2 of the insecticides evaluated were Closer, which is sulfoxaflor, a sulfoxamine insecticide with mode of action group 4C, and Savanto, or fluperidifurone, which is a butenolide insecticide having mode of action group number 4D. All of the group 4 insecticides, including the neonicotinoids, 4A, have a similar mode of action. They are nicotinic acetylcholine agonists. Courier, or buprofezin, is a growth regulator that only impacts nymphal leafhoppers while having no effect on adults. Torac, or tolfenparad, and Belief, fluonicamide, are completely different modes of action from the other materials evaluated. The insecticide treatments were applied by the farm using backpack sprayers once a week for 3 weeks. Members of the Vegetable Entomology Laboratory visited the farm to collect samples before the first application and 2 and 6 days after each application. Leafhoppers were collected using a bug vac into plastic containers, which were brought back to GCREC for processing. This figure shows the average number of watercress leafhopper adults collected from each treatment over 6 collection dates, using a 49-meter transect across each plot as a sample unit. Each treatment reduced leafhopper numbers relative to the untreated control seen along the bottom. However, the greatest reduction was produced by Courier at the top. Courier is the insect growth regulator buprofezin, which is in the mode of action group 16. Considering that buprofezin only kills nymphs, this is an interesting result and suggests there was limited migration of adult leafhoppers from plot to plot. The results of this trial were used to apply for new insecticide registrations for some materials for watercress, and to help watercress growers design season-long management programs in response to the newly established pest and phytoplasma. The watercress leafhopper is an example of a relatively obscure pest that arrived and required a response. Bemisia tabassi, the silverleaf whitefly, is a well-known pest of many crops in Florida. Up until the late 1980s, the variant of B. tabassi in Florida caused little damage. In the late 1980s, the silverleaf whitefly became established. It transmits many viruses, including tomato yellow leaf curl virus, and induces disorders, including irregular ripening of tomato and the silvering of cucurbit leaves that gives it its common name. In 2016, another variant of B. tabaci, the Q biotype, was determined to be established on landscape plants in residential areas in many parts of Florida. These different variants are presently considered to be distinct species. However, for clarity's sake, I will refer to them as biotypes in this presentation. Both the silverleaf whitefly and the Q biotype whitefly develop resistance to insecticides, but the Q biotype has demonstrated the ability to develop much higher levels of resistance than the silverleaf whitefly, or B biotype. In other countries, the Q biotype has demonstrated high levels of resistance to certain insecticides commonly used to manage whiteflies in Florida, including insecticides listed in this table. The Q biotype has consistently demonstrated the ability to develop high levels of resistance to the neonicotinoid insecticides imidacloprid, which is Admire, and thiamethoxam, which is sold as Platinum and Octara. Another factor that determines the likelihood that a new arthropod will become a pest is its ability to compete with existing arthropods. Research from China shows that the silverleaf whitefly outcompeted the Q biotype on tomato, cucumber, and cabbage, but that the Q biotype outcompeted the silverleaf whitefly on pepper. Most of our information on the Q biotype whitefly comes from other countries. This information indicates that when insecticides are overused, the Q biotype becomes resistant and can displace the B biotype or silverleaf whitefly, even though the silverleaf whitefly has biological advantages over the Q in the absence of insecticides. It's important to keep in mind, however, that the Q biotype whitefly has been detected in more than half of the states in the US, but has never caused significant problems anywhere in this country. In 2019, silverleaf whitefly populations were brought back from commercial farms from across South Florida and tested for susceptibility using a maximum dose bioassay that tests the top labeled rate of the insecticide on whitefly adults confined in a petri dish with cotton leaves that have been treated with the insecticide. The insecticides tested were the commonly used systemic insecticides in the Group 4 and Group 28 mode of action categories. These are the insecticides commonly used in the early stages of tomato production to protect against whiteflies and whitefly transmitted virus. We added bifenthrin, a commonly used pyrethroid insecticide, for reference. This map shows the areas from which whitefly populations were collected for testing. They were tested at GCREC along with laboratory colonies of a susceptible silverleaf colony and a population of the Q biotype that had been collected in 2017 from hibiscus at a residence in West Palm Beach. This graph shows the results of the maximum dose bioassay testing for 2019. The colored lines show the percentage of surviving whitefly adults from different populations under different insecticide treatments. The different populations tested are arranged horizontally across the bottom of the graph, with the Q biotype population on the far left, the field collected populations of silverleaf whitefly in the middle, and the laboratory colony of silverleaf whitefly on the far right. The lab colony of silverleaf whitefly is highly susceptible to all insecticides. The black line along the top is the survival in the untreated control. The higher the line, the more whitefly survived, and the lower the efficacy of the insecticide that was tested. You can see that with few exceptions, the red line is the highest line. The red line is imidacloprid or Admire, which consistently offered the weakest control of whiteflies. You can also see that the bright blue line is usually the lowest line. The bright blue line is dinotefuran or venom, which tended to offer the best control. The populations are arranged with the most insecticide tolerant on the left and the most insecticide susceptible on the right. The Q biotype population, which was tested in 2019 and again in 2020, was the most tolerant, but this tolerance was mostly to imidacloprid and thiamethoxam, that is, Admire and Platinum, as has been observed in other parts of the world. Here we've put a box around the response of the Q biotype population to the insecticide treatments when it was tested in 2019. You can see the blue circle, which is survival under thiamethoxam, is right below the black circle for the untreated. The red circle denoting survival under imidacloprid is beneath the blue circle, so you can't see it. This means that Admire and Platinum had essentially no effect on the Q biotype population when tested 2 years after it had been collected. Treating the Q biotype with Admire and Platinum was the same as not treating them with anything. The good news is that venom and the other insecticides tested did demonstrate efficacy against the Q biotype. Growth room studies were carried out to determine how well the Q biotype population collected from West Palm Beach competed on tomato, pepper, and watermelon with the silverleaf whitefly. There were 3 treatments. Some plants received only silverleaf whiteflies. Some plants received only Q biotype whiteflies. Some plants received male and female silverleaf and Q in equal numbers. Their development was tracked over 3 generations. Here you can see what happened on tomato when equal numbers of male and female silverleaf and Q biotype whiteflies were placed on individual plants grown in cages in a growth room. The solid black line shows the number of silverleaf whitefly females. The broken black line shows silverleaf whitefly males. The red lines are Q biotype males and females. By the 3rd generation, the silverleaf whitefly had largely outcompeted the Q biotype and was predominating on the plant. These graphs show similar results for competition studies on watermelon and pepper. The orange shows the proportion of the silverleaf whitefly, here referred to as MEM1, and the blue shows the proportion of the Q biotype, referred to here as MED. Our results show the silverleaf whitefly consistently outcompeting the Q biotype in the absence of insecticide applications. The results on pepper are different from what has been observed in China. The subgroup of Q biotype in China is different from the one commonly found in Florida. These results underline the importance of collecting local data and not relying on research from other regions to predict how new pests will behave in Florida. In closing, we want to mention a newly arrived pest of leguminous crops, the Asian bean thrips, Megalorothrips ucitatus. The Asian bean thrips was detected in March 2020 by Glades Crop Care and is presently causing significant damage to snap bean fields in Miami-Dade, Hendry, Collier, and Palm Beach counties. It is a large black thrips with banded wings. It is orange or pinkish in the larval stage. As discussed, step 1 with a new pest is to determine its host range in Florida. In addition to snap bean, Asian bean thrips has been detected on leguminous weeds and specialty legumes, including pigeon pea and lablob bean. In this slide, you can see DPM student Andy Jean-Louis and postdoctoral researcher Bruno Rosito de Marchi collecting Asian bean thrips from a perennial hedge of pigeon pea in Homestead. The crop to the right in the distance is lablob bean, also highly infested with Asian bean thrips. This graph shows the results of a maximum dose bioassay for Torac, XRL, Lanate, and Radiant against 3 populations of Asian bean thrips. Torac is not presently registered for use on legumes in Florida. Based on lab bioassay results, Lanate, Radiant, and XRL have efficacy against Asian bean thrips. Presently, Asian bean thrips has not been detected north of Lake Okeechobee. It is considered a pest of tropical growing environments, but we don't presently know how far north it will establish. If you are interested in participating in monitoring and insecticide screening of Asian bean thrips, please contact Craig Fry of Hendry County Extension. If you have any questions about the research described in this presentation, please contact me using the phone number and email listed. Thank you.